Hello students, today we are studying about budding. Budding is the process in which a small projection or outgrowth called bud arises from the parent body which matures and separates as an independent daughter individual. Now let's see, this is a parent body. In the parent body, there is a formation of a small outgrowth or the projection that is called as bud. This bud then gradually grows, matures, and separates out from the parent body and lives as an individual daughter cell. Budding are of two types. One is exogenous or external budding, and another is endogenous or internal budding. In external or exogenous budding, there is a formation of an outgrowth or bud on the external surface of the parent body. For example, in yeast and hydra. This is an example of yeast wherein this is vacuole and this is nucleus. Now, in yeast, there is a formation of small outgrowth from the parent body which gradually grows and matures and separates out from the parent body and lives like an independent daughter individual. Same happens in the case of hydra as well. Now, one more example is of cycon. In cycon, there is a formation of a growth. In yeast and hydra, the buds detaches from the body of the parent. But in case of cycon, small buds are formed and these buds later results in the formation of a daughter individual, but these individuals are attached to the parent body. But in case of yeast, it is detached from the parent body. So in cycon, the outgrowths or the daughter individuals remain attached to the parent body and forms a network and lives together as an individual. Now, in case of endogenous or internal budding, there are in this budding, the buds are formed within the parent body. For example, in case of spongilla or sponges, on unfavorable condition, the buds are formed within the body of the sponges, and each cells are called as archaeocytes. These archaeocytes are collectively called as gamule. So these internal buds are also known as gamules. Okay, now these gamules comes out from the body of the sponges on the arrival of favorable conditions. On arrival of favorable conditions, these gamules or the archaeocytes comes out from the body of the sponges and later lives like a daughter individual. Apart from endogenous and exogenous internal budding, there is also one type of budding called strobilation. What is strobilation? Strobilation is the process in which the repeated formation of similar segments detaches from the body of the parent organism. It generally occurs in Aurela. Aurila. Aurela. Aurelia. Aurela. In Aurela, the body separates out from these segments and each segment is capable of forming the new individual or the daughter individual. These segmented bodies are called strobila and the process is known as strobilation. These segmented bodies are called strobila and the process is known as strobilation, where there is the repeated segmentation and each segmentations are also known as epiphyra. wherein one epiphyra leads to the formation of one individual daughter cell. Next is fragmentation. In fragmentation, parent body breaks into two or more fragments, and each fragment then develops into a new individual. For example, in case of nostoc and Spirogyra, both of them are the algae. In Spirogyra or nostoc, this is a parent body from which the daughter cells have parent body have been broken down into two or more fragments, and these fragments then develops into a new organism and starts living independently.